Hi, my name is Santhoshi Krishnan, and I'm a fifth-year doctoral student in the Department of Electrical and Computer Engineering at Rice University. In this talk, I'll be highlighting my paper named GaWRDenMap, a spatially informed framework to quantify heterogeneity in intercellular interactions in the tumor environment. There are a variety of therapeutic and palliative measures used in cancer treatment and management. Of all of these, immunotherapy is gaining a lot of prevalence in the past few years. Briefly put, in this method, certain parts of the person's immune system is mobilized to fight diseases such as cancer by stimulating and boosting the natural defenses of the immune system to work harder and smarter to find and attack these cancer cells. Unfortunately, only certain cancers, such as pancreatic and melanoma, have been shown to be responsive to it so far. In the case of other therapies, the treatment paradigm is more streamlined and generalized, which may lead to unintended side effects and may not work the same way for everyone. This is because there is a difference in the way the disease manifests itself between patients due to variation in the individual response to environmental and innate genetic factors, but also a difference present within the same patient. The tumor microenvironment consists of a myriad of cells which are interacting with each other in different ways, and their interactions in a functional manner is dependent on their minimum threshold of distance from each other as well as other types of cells. Thus, there is an increasing emergence of the need to characterize the tumor microenvironment in a spatially sound manner. As an example, if you look at these different slides represented with the red representing immune cells and the blue representing tumor cells from different patients suffering from the same disease, though we may assume that the proportion of both the red and blue cells might be similar, there is a marked difference in the spatial arrangement of the cells, which in turn informs the functional effectiveness of each of these cells on each other. We observe that the cells are well separated in the sample in the blue box. In contrast, the cells are well intermixed in the case of the sample in the red box. Keeping this in mind, we want to develop effective ways to capture and differentiate between these different kinds of interactions. This is why we want to create a mapping of regions which are showing strong and weak mixing of phenotypes in each tissue region of observation, and we hope that this would be a surrogate for exploring functional relationships between different cell phenotypes in each of the subregions of the tissue under observation. To inform these analyses, we propose the GaWRDenMap framework, which utilizes the concept of geographically weighted regression borrowed from ecology and combines it with a density-based classification approach to differentiate between different cohorts of disease histologies. Very simply put, what we do over here is utilize gel point pattern density maps obtained from information from MIF stained images or any form of tissue data which has spatial information available, perform regression on this data using these surfaces as predictors and response in a spatial regression model, which in this case we selected it to be the geographically weighted regression model, and then utilize the generated regression values obtained to generate features which are then fed into the classification model to differentiate between different patient cohorts. To test out our framework, we applied this on a cohort of multiplex immunofluorescence images from patients suffering from 6 different types of pancreatic conditions, which included chronic inflammatory conditions, precancerous lesions, and adenocarcinoma. 2 cell types were considered for this study: the epithelial cells, which were cancerous in nature, and a combination of 2 types of immune cells, which were exhibiting either CD4 or CD8. Of special interest is the categorization of CP and PDAC, as both CP and PDAC are characterized by a dense inflammatory stroma composed of fibroblasts and immune cells, making diagnostic discrimination on biopsy difficult. So this was of a special interest to us. On looking at the classification results, we find that our framework is able to discern clinically relevant pathologies with a small selection of the results presented here. The framework can distinguish between CP and PDAC with an AUC threshold of 0.75 set as advised by a clinical collaborator. This is significant because, as mentioned earlier, both CP and PDAC are characterized by a dense inflammatory stroma, making diagnostic discrimination on biopsy difficult. In order to confirm these results, as this particular binary classifier was of special interest to us, we also applied the same classifier on a small but separate validation cohort. We observed a drop in the AUC to 0.71, but the high sensitivity and specificity values were quite encouraging. We do plan to validate some of these findings on a larger and more balanced cohort. We not only wanted a framework to be able to discern between these groups well, but also point to differences in a biologically relevant manner. As you can see, there is an evident visual difference in the distribution of the density curves between the two groups of interest. The observed greater range of coefficient values observed through our analysis suggests a greater spatial heterogeneity between lymphocytes and epithelial cells across samples in PDAC as compared to CP. Eventually, we want to use these generated GWR densities as a potential novel biomarker or an interaction signature in such similar cases, where standard pathological analysis can lead to ambiguous conclusions that are frequently encountered when distinguishing pathologies with overlapping differential diagnosis. Additionally, though in our case, we had only queried 2 different cell phenotypes, it is possible to extend this framework for 3 or more different phenotypic surfaces. One such example would be to investigate the change in 3-way interaction between tumor, CTLs, and Tregs across the cancer microenvironment. Additionally, this method is not restricted to just multiplexed immunofluorescence data or any kind of imaging data. It can be extended to any application where spatial information and phenotypic information is available, for example, in applications of spatial transcriptomics. I would like to thank the following people for helping me in supporting this project. Thank you.